Okay, my conflicts, as they're disclosed, I'm also discussing investigational drugs. So, in psoriasis, I believe today we're really at a break point where we are able to practice molecular medicine in a very sensible way to treat this disease. And essentially what we have done over a course of about 15 years is elucidate elements of pathogenesis in human cells and tissues that have created hypotheses which have been tested through targeted therapeutics. In each of these studies, because we have an accessible organ, we've learned something which has led back to the understanding, and we have gone through several iterative cycles of this to the point today where in the last decade there have been 6 new approved drugs for psoriasis, 1 withdrawn because of a PML issue associated with it, but then extremely broad current testing of new agents in trials which are largely focused on the IL-23 Th17 pathway, although some agents are broader than that. So what I want to do is introduce you to this disease and to the pathways of drugs and discovery. So I think the case can be made that psoriasis vulgaris is perhaps the most successful case of molecular therapeutic targeting in a complex genetic human disease. We now have drugs, as I will explain, that directly target inflammatory elements that are part of the genetic risk of the disease, and there has been an extremely rapid evolution of both our pathogenic understanding and therapeutic development in the last decade. However, I might be the only dermatologist in the room, and therefore I think most of you would ask, well, what is psoriasis? So, psoriasis vulgaris is an inflammatory skin disease which is characterized by focal skin lesions. They're red on their base, they're raised up relative to adjacent skin, and they have this stuff that we call scale that kind of looks like you've scraped your knee on the floor, and you would call this a scab if it occurred on a kid after a playground accident. So if one does a biopsy of this area versus the psoriatic plaque, you get this kind of picture in which there is a normal epidermis composed of keratinocytes. In non-lesional skin, it sits on a base that's pink of collagen with connective tissue cells and a few immune cells in it. The psoriatic lesion, as you see at the same magnification, is this tremendous growth of the epidermis. It's thicker, it has downward protrusion we call rete, it has cells with retained nuclei that we call parakeratosis, and it sits on a base in which there are many mononuclear inflammatory cells present. Now, essentially, there are 2 invariant features of this disease. One is that the epidermis is proliferative. So one can view the level of proliferation by Ki-67 staining for cycling cells. Normal skin, 1 in 10, 1 in 20 basal keratinocytes in cycle. The psoriatic lesion, in contrast, virtually confluent cycling of basal keratinocytes, some piled up suprabasally. And with this growth activation, there's a differentiation shift into a wound healing program which is conveniently marked by Th17 expression, so you can see Th17 positivity here, and not in suprabasal cells in normal skin. So this is just a very convenient marker of this growth turned on state. Now, the other thing that is invariably associated with the psoriatic phenotype is tissue infiltration by mononuclear leukocytes, of which T cells are a very prominent subcomponent. And so what you can see in the psoriatic lesion are these perivascular aggregates of T cells, along with scattered T cells in both the dermis and some infiltrating T cells in the epidermis. These here are mostly CD8 positive. These are mostly CD4 positive. Normal skin does contain a complement of T cells, but much lower and virtually none in the epidermis. Now, the way these 2 invariant elements of psoriasis go together in terms of pathogenesis is a model that was put forward around 1995 that was based on therapeutic targeting of the activated T cell with an IL-2 containing toxin. And this model suggested that there was some critical interaction between a growth-activated T cell and a keratinocyte that drove a resting epithelium into this regenerative or wound-healing mode, and associated with this was not only the thickening that I showed you, but also the expression of certain immune adhesion molecules on keratinocytes that could be induced by molecules like gamma interferon and TNF, and which would then promote this adhesive interaction. Now, the T cell model was created on the basis of some small experiments experiments that were done with this fusion toxin, but it was fundamentally confirmed by non-cytotoxic agents that followed CTLA-4 Ig, a molecule called lefosept, and evolizumab, which targets LFA1. Now, the model that we put forward is tremendously out of date and needs to be updated to include elements of immunity that have been discovered since this model was put forward. So it was built on the idea that there were only Th1 and Th2 T cells, and it now needs to be updated to talk about Th17 and Th22 T cells. We now have a prominent role for dendritic antigen-presenting cells in the process, and so that needs to be updated. And the model provided no molecular mechanism that could link hyperplasia with the presence of immune cells, except as an injury response. And so now we can update that to talk about specific cytokine effects on keratinocytes that may give us the hyperplasia and patterning. So one version of the updated pathogenic scheme of this disease is this diagram. What it suggests is that we have a dendritic cell that we call a TIP-DC, which is an immature DC that can mature towards a more mature antigen-presenting cell, and that this dendritic cell then drives the activation of Th1, Th22, and Th17 T cell subsets, where the cytokines that define the T cells of each subset are produced, act on keratinocytes, which have receptors. The output of this at the keratinocyte level is the production of many other inflammatory molecules, some of which feed back and reinforce this immune reaction that can occur perpetually in skin unless it's interrupted with a therapeutic. But out of this more complicated set of reactions, the thing I want to make an argument for is that this disease may reduce to mainly a disease of TIP-DCs producing IL-23, activating Th17 T cells, and with unique keratinocyte responses that set up our visual and our molecular definitions of this disease. So let me start with elements of this model. I want to start with dendritic cells in skin. There are two kinds of dendritic cells in normal skin. They are Langerhans cells in the epidermis, marked by Langerhans or CD1A, and dermal dendritic cells, shown here in green, which are marked most efficiently by the integrin CD11C. Now, in psoriasis lesions compared to non-lesional skin, what you can see consistently, as I've told you, is an increase in T cells in lesion, and that's shown here by cell counts, comparing non-lesional to lesional skin. Likewise, there's a very consistent increase in dermal dendritic cells marked by CD11C in this disease, without much change at all in Langerhans cells. And this increase in dendritic cells over the background number in skin led us to ask, is there something different about these inflammatory DCs? And with that, we started to look at markers of these DCs and found that TNF and INOS were highly consistent molecules associated with the DCs. So that if one stains tissue sections for TNF and CD11C here separately, you can see the convergence image at the bottom is that in the dermis, almost all of the TNF is associated with these myeloid dendritic cells. Likewise, there is a dendritic cell staining pattern for INOS, which in two colors would put INOS in the myeloid dendritic cell. And so we called the cells in psoriasis TIP-DCs for TNF and INOS production in parallel with the discovery of a cell called a TIP-DC in mouse 1 year before that was discovered in the spleen during Listeria infection. But both of these cell groups then have the idea that they may carry some innate effector functions forward, as well as having traditional roles as antigen-presenting dendritic cells. So we initially called this the TIP-DC, but have gone on to study other molecules that are associated with it. These molecules include toll-like receptors, and also secreted molecules that include S100A12, the cytokine IL-20, and perhaps most importantly, IL-23. So what is IL-23? IL-23 is one of two so-called P40 cytokines. These are cytokines that share a common P40 protein subunit. The association of P19 with P40 gives uniquely IL-23. The association of P35 with P40 gives IL-12. These bind to receptor complexes that are half related through the IL-12 receptor beta-1 chain, and then there is JAK-STAT signaling. Now, the important thing about IL-23 is that it is consistently elevated in psoriasis lesions. And so one if one compares uninvolved to lesional skin, this is a logarithmic scale of gene expression. You can see the connected lines are individual patients, that every single patient studied has increases that are parallel in both P40 and P19 subunits of IL-23. Now, why is that important? As we understand current cytokine biology, IL-12 is perhaps the master cytokine driving Th1 T-cell activation and differentiation, whereas IL-23 is the master cytokine of at least survival of Th17 T-cells in an activated state. Now, Th17 T-cells are defined by their ability to make IL-17, whereas Th1 are defined by their ability to make gamma interferon. In mouse systems, Th17 T-cells make both IL-17 and IL-22. A bit of a complication in humans that most IL-22 is made by a separate T-cell subset that we call Th22. For the purpose of this talk, I may lump together both of these in a way that's a little bit imprecise. So, are Th17 T-cells increased in psoriasis lesions? The answer is yes. So if one does individual cell phenotyping by FACS, compares blood versus skin of the same patient, what you can see here is that there are a population of IL-17 producing T-cells that are clearly increased in abundance over the population in blood, and these coexist with a population of Th1 T-cells, which are defined by the ability of these cells to make gamma interferon. Now, this method says the cells are present in tissue. It doesn't say or address whether they are activated. And I think the best way we can ask that question is by looking for in situ synthesis of the cytokines that define the activated T-cell subset. So as a control, this is Th17 expression at the mRNA level. It's dramatically increased in psoriasis lesions. Likewise, there's an increase in gamma interferon, IL-17 and IL-22 mRNA in active psoriasis lesions. Now, is the expression in any way related to ongoing disease activity? And so to answer that question, we have taken patients at baseline and suppressed disease activity with cyclosporin. You can see here Th17 decreases as a measure of the reduction in skin disease. And in a parallel fashion, you see reductions in gamma interferon, IL-17, and IL-22. Now, this simply says these cytokines might have something to do with pathogenesis. It doesn't prove that there is any relationship at all, and it doesn't address what might be the unique functions of different cytokines in creating this disease phenotype. So I want to ask what features of psoriasis might be explained by Th17 T-cell products, IL-17 and IL-22. So I want to go back to this original picture that I showed you of tremendous hyperplasia that is associated with the psoriatic phenotype. But I need to confess immediately that hyperplasia is not the only defining feature that we use to give a diagnosis of psoriasis. So as an example, I want to show you a biopsy of normal skin in relationship to two inflammatory skin conditions. Both have thickened epidermis, keratin-16 positive keratinocytes, and Ki-67 activation in keratinocytes, so they're cycling. Both of these could be biopsies of psoriasis, and I can't actually tell the difference between what they are, so I'll supply the names. This is a biopsy of psoriasis, and this is the other very common inflammatory skin disease, atopic dermatitis. Now, we've gone about trying to define what are the unique molecules associated with psoriasis that define it, distinguish it as a disease from atopic dermatitis. And in doing this, we've used gene arrays to create gene maps of the diseases. Here in the middle is the overlap of 366 genes that are shared between both diseases. And many of these genes are actually related to epidermal activation. And so in the red, you can see genes that are commonly activated in both conditions. They include Ki-67 and a group of cyclins that are related to proliferation. And so while one might have inferred that both of these are diseases of keratinocyte hyperplasia from just measuring epidermal thickness, I think probably the gene expression here is maybe comforting confirmation that our molecular probes are on the right track. So from that background, we go on to define what are the unique genes that distinguish psoriasis from atopic dermatitis. And so here we have a group of genes that are in red, that are at high expression expression in psoriatic lesions. They're not expressed at very high levels in normal skin. And in atopic dermatitis lesions, each one of these gene products is at a statistically lower level of expression than in psoriasis lesions. Now, what are these genes? Well, some of these are genes called psoriasin, discovered as a protein in psoriasis scale in the first, and S100A9. Now, where are these elements, these proteins that are encoded localized? Well, they are localized largely in keratinocytes, as you can see here, by the staining of normal versus psoriatic skin. And so one can ask, what in fact are the molecular inducers of these relatively psoriasis-specific proteins? And this is where Th17 T cells become very important. So an experiment's published about 5 years ago from Kirstine Wilk, who was in Robert Sabat's lab at the Charité in Berlin. They did a very simple in vitro experiment in which they incubated IL-22 with human keratinocytes in a culture dish, and what they found was psoriasin and S100A9 were very strongly induced by IL-22, but not by gamma interferon. And this began to set up the idea that Th17 cytokines might have unique effects on keratinocytes with respect to this disease. There was then a paper published from Wenjiang Ouyong's group, who's at Genentech, that was in Nature a few years ago. They injected IL-23 into mouse skin. It led to a Th17 T cell accumulation and to epidermal hyperplasia and inflammation that shared some features with psoriasis, and this was called psoriasiform. Now, importantly, if they did this 23 injection on an IL-22 null mouse background, most of this hyperplasia didn't take place. So there was very clearly strong dependence of the phenotype on IL-22 production. That group also published this other very neat study in which they took human keratinocytes cultured on rafts so that keratinocytes differentiate like they do in normal skin. They added in a number of IL-20 family cytokines that includes IL-22 and saw that epidermal thickness was dramatically increased by IL-22. Keratin 16 was induced, and psoriasin was also very strongly induced along with activation of STAT3. Now, all of these are either cellular or molecular features of psoriasis, and it began to set up the idea that IL-22 might be a very important cytokine in controlling patterning and growth features of this disease. Now, we repeated that experiment on a slightly different culture model, and when we did, we could also see that in addition to acanthosis, parakeratosis, or the retention of nuclei, was induced by IL-22. Now, in contrast, IL-17 induces almost no epidermal hyperplasia, so its role is less clear from this kind of experiment. Now, Th17 T cells are, of course, named for IL-17, so you can ask, what does IL-17 contribute to psoriasis? And in that way, we've repeated the same kind of experiment that was originally done by the Sabat lab with cultured keratinocytes, comparing IL-17 to IL-22 to gamma interferon. And what we have found is there are a number of molecules that are specifically induced in keratinocytes by IL-17. These include not only S100 proteins, but also CCL20 and a group of chemokines that are involved in neutrophil trafficking. And if we go back to our molecular map of genes that are highly expressed in psoriasis and distinguish this disease from atopic dermatitis, we essentially can paint the map that either IL-17 or IL-22 are the molecular inducers of these molecules in keratinocytes. So what the IL-23 Th17 pathway then potentially gives us is the idea that IL-17 that is produced by Th17 T cells can lead to recruitment of both dendritic cells and Th17 T cells via the induction of CCL20, that neutrophil trafficking chemokines are overproduced in the presence of neutrophils is a feature of this disease, and importantly, there are innate defense molecules such as lipokeltin and LL37, which may play another particularly important role in driving the chronicity of this phenotype. Michel Gillier's lab has discovered that LL37 or cathelicidin plus self-RNA has a dramatic ability to mature immature dendritic cells into mature cells which are marked by DC lamp in humans. So I think, as most of you know in the room, the bulk of dendritic cells that are in peripheral tissues exist in an immature state, and when they see TNF-alpha, other cytokines, or toll-like agonists, they mature into cells that have upregulated antigen-presenting machinery, and in humans are marked by the presence of either DC-LAMP or CD83. If we look in psoriasis lesions, there are large accumulations of DC-LAMP positive dendritic cells, which are not just solo cells, but are intermixed with T cells, and create the idea that this may be an organized lymphoid tissue that is driving chronic T cell activation in the disease. And so we've put forward that there is a molecular environment in the skin that includes upregulation of CCL19 as well as CCR7 expression by the cells that are here, and the idea that chronicity of this disease may in fact be controlled by the formation of these mature DC T cell aggregates, and also more recently by laser capture microdissection we've seen that CCL19 is actually made by the cells in these aggregates, so this may be the equivalent of T cell zones of lymphoid tissue in the skin. So our updated model for pathogenesis is the idea that we have these dendritic cells. They are potentially directing Th17 and Th1 activation by making 23 and 12. Cytokines that are produced by activated T cell subsets act on keratinocytes to set up unique molecules that are controlled by these cytokines in the disease pathogenesis. Now, all of this makes for a pretty good story, but I haven't given you one shred of evidence that this is actually an important pathway in pathogenesis. So you might ask, where's the beef? Well, I think the beef is provided by two therapeutic antibody antagonists which have been tested in psoriasis patients. They are P40 monoclonal antibodies that are called ustekinumab or ABT874 or breakinumab. They bind to the common P40 subunit shared by IL-23 and IL-12 and neutralize the cytokines. And so one can ask, in the background of this model, are these elements actually correct as drawn? So ustekinumab is a antibody that hit a home run with its first safety study in humans. This is a test of the antibody by Kaufman et al. And what they showed was that a patient who received a single dose of this antibody at baseline 16 weeks later had completely clear skin. And this, of course, launched proper phase 2 and phase 3 studies, which have culminated in 3 phase 3 studies that are published with ustekinumab. 2 of these are placebo-controlled, and 1 is a comparator study with etanercept. And the results are really very, very clear, because psoriasis as a disease has a very minimal placebo response, meaning that the disease is remarkably stable in the skin unless treated. So 3 or 4% of patients in these phase 3 trials had a PASI 75 response at week 12 versus between 60 and 70% of patients that were treated with either 45 or 90 milligrams of ustekinumab given twice. Now, another remarkable thing about this antibody is the stability of the response when somebody is on continuous treatment. So these are now data out to 2+ years, which show that once one attains a PASI 75 response, in large part that response is maintained with treatment over a period of 2 to 3 years. Now, this study, the ACCEPT study, was a randomized comparison trial between ustekinumab and etanercept, and it's within this trial that we were able to do a number of mechanistic tests in conjunction with Carrie Brodmerkel's group at Synacor for essentially trying to determine whether our cytokine models were correct in terms of the cellular output. So the first prediction of the model is that blocking IL-12 and 23 should decrease gamma interferon and IL-17 output from T cells, and that's in fact what happens in 12-week treated lesions. IL-22 is also reduced as predicted from the model. Now, perhaps not predicted, there appears to be feedback such that IL-23 itself is reduced, and INOS, which is a gene of the TIP-DC, is also very strongly attenuated. So multiple elements that are upstream on this model are blocked. So blocking IL-23 and 12 decreases output of IL-17, 22, and gamma interferon. And the next prediction of this model is it should have a major effect on both the activation and the molecular phenotypes of the cells that are used to define psoriasis. And that is looked at in this trial by a combination of histology and and molecular probes. So this is baseline psoriasis before treatment, and here is the same plaque at 12 weeks of treatment. You can see that we've reversed hyperplasia about back to the level of non-lesional skin. Keratin 16 is negative in suprabasal keratinocytes. Infiltrating T cells and dendritic cells are reduced about back to background levels that are seen in a non-lesional skin. So there is a major reversal of the phenotype, and essentially once one attains a PASI 75 or a PASI 90 response, this reduction from thick epidermis to thin epidermis is very, very consistent across different patients that are treated. Now, the other way that this has been looked at is by gene arrays, in which one can define several thousand genes that are different between uninvolved and lesional psoriasis. And so here are slightly more than 2,900 genes which are either upregulated in psoriasis plaques at baseline or downregulated in psoriasis plaques relative to non-lesional skin. And what this shows you is that these set of altered genes in the disease are normalized by treatment for 12 weeks with either ustekinumab or etanercept. So there is high-grade reversal of the molecular phenotype of the disease, and essentially all of the individual elements that I'm painting here as induced by IL-17 or gamma interferon are very highly suppressed by blocking the primary cytokines. So now we have a bit of a therapeutic puzzle. So if IL-23 and Th17 T cells really drive this disease, how can we treat psoriasis with TNF antagonists, particularly if you view TNF as a master cytokine of innate immunity? So within the model that I have, one could say, well, TNF could play its classic and most understood role as the master cytokine of the sepsis cascade, the idea that TNF induces IL-1, IL-6, IL-8, and all of these are elements that are in fact elevated in psoriasis lesions, so that could be true. But instead I want to make the case that this is not the critical pathway of TNF in this disease. It's in fact this, the idea that TNF interacts with the myeloid dendritic cell and that TNF drives IL-23 production, which then drives the Th17 pathway as the most important element of pathogenesis. And the data for that are a small study that we conducted with etanercept. So we essentially did biopsies at baseline, 1, 2, 4, and 12 weeks in patients that had clearing of their skin under etanercept treatment. And what we saw is that within 1 week of starting treatment with etanercept, 1 to 2 weeks, there is really marked reduction in IL-23 production in the skin, and you can see here how the other markers of the inflammatory dendritic cell are being reduced by blocking TNF. So this prevents the TIP-DC from expressing some inflammatory products, but perhaps more importantly, it reduces synthesis of IL-23, and thus should have a dramatic impact on Th17 activation. And in fact, one can measure that by looking at IL-17 levels in skin. 1 week after starting TNF blockade, there is a dramatic reduction in IL-17 production in psoriasis lesions. And at the end of 12 weeks of treatment, the extent of disease improvement, as measured by a combination of clinical and histology measures, is very highly correlated with something that we call the Th17 score. The Th17 score is a group of downstream genes that are regulated by IL-17 of the kind that I have shown you. Now, what about the difference between responding and non-responding patients? Well, if we look at all of the patients in the study, I have here shown you or plotted the cytokine inhibition profiles of IL-1 and IL-8, which are these sepsis cascade genes in both responding and non-responding patients. And I think you can see that the extent and kinetics of suppression are essentially the same whether one has a responding or a non-responding patient with psoriasis of the skin. Instead, the thing that appears to control response is suppression of this group of IL-17 pathway genes in responders, and there's very little movement in this pathway in non-responders. So I would argue that while one might have conceptualized TNF as a cytokine primarily regulating innate immune responses, in fact the data in psoriasis suggests that this is a much more invasive kind of cytokine, and in fact its biological actions extend into the same biological space where IL-12 and IL-23 control inflammatory reactions. Now, We're back to this model, which I suggested might be some of the central elements of psoriasis. I want to just highlight that for our TYP-DC, blocking TNF has major effects to downregulate this pathway downstream. INOS, as it turns out, is actually a risk gene by GWAS studies for the inheritance of psoriasis. Both subunits of IL-23 and the IL-23 receptor contain risk alleles for the genetic transmission of psoriasis. So these are genes that are tightly linked to the acquisition of the phenotype. And in the case of the IL-23 receptor, the risk allele appears to control more Th17 differentiation from naive T cells. Now, further down this pathway, there is IL-17 as an inflammatory cytokine, where there are two studies that have been done now with antagonists that suggest that this cytokine is very important in controlling the psoriatic phenotype. Now, one of these studies is a study that was published from Huber et al. In the Journal of Translational Medicine with AIN 457, Novartis' anti-IL-17A monoclonal antibody. You've already heard about the results in rheumatoid arthritis. In the case of psoriasis, the injection of a single IV dose of this led to a PASI 75 response in about 40% of patients after 4 weeks of treatment. Now, this is about the same level of PASI response that's seen with etanercept after 12 weeks of treatment. So already this is a remarkable result, and trials are in place now testing multiple doses of the antibody for the clearing of psoriasis. There are two other anti-IL-17 programs. Eli Lilly has also an IL-17A antibody where the results have not yet been made public. And Amgen has an IL-17 receptor antibody that is now in phase 2. There's a concept study that was presented a year ago at the SID that I want to review some data in, and a phase 2 study that was very, very striking that was reported 2 weeks ago at the World Congress of Dermatology. So let me tell you just very briefly about this inhibitor, an antibody called AMG 827. It binds to the IL-17 receptor A and blocks signaling by IL-17A, IL-17F, and IL-25. And when this was given IV at high doses to psoriasis patients in a proof of concept study, I just want to point out here that at the highest dose tested, 7 of 8 patients had a PASI 75 response at the end of the study. And if you look at the kinetics of this, many patients actually had dramatic improvements in the PASI score at only 2 weeks of treatment. Now, this is seen clinically by typical psoriatic plaques by day 15 that have largely melted away, and by 6 weeks of treatment, the skin looks virtually normal except for a little bit of post-inflammatory hyperpigmentation. Now, while that is very striking, the biopsy and molecular results that have come out of this study are even more striking. So one can see that by only 2 weeks of treatment, the proliferation of keratinocytes is essentially turned off, and we have levels of proliferation that are back down to baseline level. And by 6 weeks, we have epidermal reversal back into the normal homeostatic growth pathway, much like non-lesional skin. Now, perhaps even more impressive than this histology results are the effects of blocking IL-17 on gene expression. So these are data that come from Chris Russell, who is at Amgen, and Chris is a master of doing gene arrays, much more so than we are. And what he has shown here is about a group of 10,000 genes that are elevated in psoriasis lesions compared to normal skin. So this is a volcano plot that's put horizontally. So this is the tip of the volcano over here. The blue is gene expression in non-lesional skin, and the light blue and the green are genes that are different in the psoriatic lesion. And you can see here this is a logarithmic scale of expression on a log 10 scale. So there are a number of gene products here that are elevated by about 10-fold that include primary cytokines along the Th17 pathway that we have been discussing, as well as many other genes that set up the molecular definition of the disease. By only 2 weeks of treatment, what you can see here is this 10,000 gene signature is reduced to a gene signature of about 500 products that are elevated in psoriasis without much further movement as one goes out to 6 weeks of treatment. So essentially, the disease has been turned off by 2 weeks by blocking IL-17. Now, how can IL-17 so rapidly shut down what are hundreds or thousands of genes within only 2 weeks? In the first set of experiments that we did with IL-17, its inductive effect on gene expression in human keratinocytes was only about 40 genes as measured by the same series of arrays that Chris has used. And while he's better at doing gene arrays than we are, it's nothing like the difference between 40 and 10,000. So is there another explanation? Well, I think the explanation comes at the level of the keratinocyte. For the cells that express IL-17 receptors most abundantly in human skin are, in fact, keratinocytes themselves. And so the idea I want to put forward is that there is a critical interaction between IL-17 and TNF at the level of the keratinocytes that turns on a whole set of key and highly inflammatory genes in this cell type. And the way we have gotten to that is by, again, a very simple in vitro experiment in which we studied individual cytokines and mixtures of cytokines that include IL-17 and TNF added to cells at the same time. And in this, we have seen that there are literally hundreds of genes that are regulated in additive or synergistic fashions by these two cytokines. Now, I realize that this table is nearly impossible to read from anything beyond the first row, so I just want to point out at the top I'm showing IL-19 expression. Individually, IL-17 or TNF induce expression of this IL-20 family cytokine by less than 2-fold, but the induction by the combination of IL-17 and TNF is more than 100-fold in synergy, and this kind of synergism or additive effect holds true for many other inflammatory genes that are key in the development of psoriasis, including IL-23 itself. And I want to point out that this set of synergistic genes that we identify in this fashion, in fact, are a major part of the transcriptome of psoriasis compared to atopic dermatitis, which is here. So essentially, these are all elevated in different gene expression studies of psoriasis, but not in a gene expression study of atopic dermatitis. So I think that would make psoriasis largely a disease with emphasis of the Th17 axis from TIP-DCs and IL-23, leading to the output of inflammatory cytokines that feeds back into this pathway. And so from that, I think we're now fairly close to our end inflammatory dissection of this disease, and we've gone through iterative cycles of hypothesis testing and therapeutic testing, which hopefully now net us with a group of new therapeutics that I expect to make their way through phase 3 trials and hopefully to new approved therapeutics for the disease. So this is my group at Rockefeller University that has been working on psoriasis for the last 15 years, although people have changed. I want to acknowledge key collaborators, Carrie Brodmarkle and Catherine Lee from the Centocor Biomarker Group. I think today Centocor is being renamed Janssen Pharmaceutical, and Chris Russell and David Martin from Amgen with the IL-17 receptor antagonism project. Thank you.
There's definitely time for a few questions. Jim, I was fascinated by the data that TNF actually may have a direct inhibitory effect on these TIP-DCs in terms of IL-23. Is this a direct effect on gene expression, or can TNF antagonism perhaps delete a population of TIP-DCs that are in skin lesions? Do you know that?
All right, so if we do the in vitro experiment where we make DCs from monocyte precursors, then the effect is either adding TNF or inhibiting TNF with etanercept leaves intact cells that specifically downregulate IL-23 and some other inflammatory cytokines. So I don't think it's death. I think it is blockade of signaling that's TNF mediated.
Great talk, Jim. The TIP-DCs, do they evolve in the skin from monocytes that have gone in there, or have you looked in the circulation? The other question related to that, what about the lymphatics? I know that they're greatly expanded in psoriasis. Is there an interaction there?
Okay, let me take them one at a time.
Okay.
The TIP-DC is elusive in its origin. In humans, we haven't been able to make it from anything from a monocyte by adding cytokines. And there isn't a circulating population that is exactly equivalent to it. In mouse systems, it's believed that they mature very, very rapidly from monocytes within a peripheral tissue environment under the influence of the cytokines that are there, and that may very well be the case in psoriasis. There's a subset of TIP-DCs that are marked by another molecule where that can be found in the circulation, so maybe there are some circulating precursors that can find their way into the skin, but the overall numbers are just huge in the skin relative to what's in blood. With respect to lymphatics, so in psoriasis, there's tremendous angiogenesis. So you see these corkscrew vessels, pretty much like you do in psoriatic arthritis joints, but the lymphatics themselves are actually collapsed. And that's something we've published in something that we call a molecular scar of the disease. But if you think about this, once one has, let's say, lymphoid tissue with structure in the skin, you may not need open lymphatics to have leukocyte trafficking back to lymph nodes. And so what we see is there's no lumen. There are only two opposed lymphatic endothelial cells in active lesions. And surprisingly, when the disease gets better, we don't see these things open back up. So that may be part of restraining leukocyte trafficking out of the skin.
Has anyone looked at, in the animal models or in humans, at the lymph nodes draining the skin?
Well, humans it's tough because we don't go around doing lymph node biopsies, so that's mostly unexplored. In mouse models, there is no mouse model of this disease, okay? Let me just say that everything is an approximation of some feature of this disease clinically, but you really can't study that very well.
Yeah. Okay, thanks.
So you showed some really very nice data on the efficacy of a couple different approaches for treating psoriasis. I was wondering if you could also comment on the safety profile of those different strategies.
Sure. So this is a chronic disease. I probably didn't emphasize adequately that often it comes up in teenage years and will persist until someone dies. So this could be a disease of 60 years. To treat a disease over 60 years, one needs something that's reasonably safe. So far, the P40 antagonists with 4 years of safety look fairly close to the signals that were seen in periods of placebo treatment. And so there hasn't been an evolving picture of toxicity over time with those. And there are, of course, human knockouts of both P40 and the common receptor, IL-12 and 23. Those individuals with a developmental defect in this pathway have increased susceptibility to mycobacteria and to intracellular organisms like Salmonella. So, that's probably the worst case for what would happen with antagonism of the pathways in adults. TNF antagonists, a little bit of an increased risk of infection with bacteria, much like what's been seen in RA, but probably of a lower order of magnitude than what was seen in RA patients.
Jim, could you speculate whether specific IL-23 blockade is going to be as good as IL-12?
All right, so there are two phase 3 trials that have been done. The results aren't public, but keep your ears open. I think the fact that the IL-17 antagonism works would predict some efficacy.